stop me at any time if you have any questions. And after that, I want us to start, I'll open Word and I want us to start writing the methodology for the assignment you will have to handle. I know for you considering you all didn't do it, I think it's ideal that I help you all do the methodology. So that's good. Sampling techniques. Entomology. So entomology is the science of insects, right? And as such, people who study it are called entomologists, just like people who study biology are called, so I think this is my applications. Anyway, just like people who study, let me make sure my mic is on, sorry about that. Just as people who study biology are called biologists, people who study, I don't know, what does they study? People who study parasites are parasitologists. People who study, what else people study? I know people who are parasitologists. Some people if you study ecology, you're an ecologist, just like that. If you study entomology, you are called an entomologist, right? So entomologists study a wide variety of topics. So I have not always been very interested in entomology, but I have a lot of friends who are entomologists, and the stuff they do is super cool. Oh my goodness. Did you know that a lot of the time, people who do forensic science, they are also forensic entomologists. Not all forensic scientists are entomologists, but they are a particular group of people known as forensic entomologists. I have a few friends who are forensic entomologists. What they do, if a body, let's not think morbid. Okay, let's say you realize that your pig, you lost a pig and then you discover the pig dead. You can see what insects are consuming the body to determine how long ago the person died. And people use that all the time in forensic science. People use that and forensic entomologists, so they can use the insects colonizing the body to determine how long ago someone died. So entomologists do a lot of things. So sometimes they study how insects interact with each other. Some insects are carnivorous, so they eat other insects. They have to study that. Others study how insects live and reproduce because we don't know much about them. So let's say suddenly we discover that there's a particular insect inhabiting and destroying farms. So entomologists have to collect that insect and probably put them in another controlled environment and see what exactly it is how are they reproducing? When do they reproduce? What medium do they reproduce in? Do they need water to reproduce? Do they, So it's a lot that they can study. They study how to keep the insects away from people's food and people's crops. And then they meet and they share ideas. And it's really interesting. As I say, I was always interested in insects, but then when you hear some of the entomologists talk, you see how fascinating the field is. So usually in entomology, you use what is called sweep net sampling. And we just showed the video yesterday. So if you can see, this is the net of what you use. So this is a lot of times they also use this to catch butterflies. So animals that are in the air. However, you could also sweep it back and forth like in your crops. So if you see this farmer right now, he is sweeping it back and forth in his crops to determine what pests maybe plaguing his crops. So when you do that, you could collect them and you can observe, okay, well, I realize that this type of horn beetle tends to eat my lettuce usually. So you try to find some way to decrease them. So when you are doing sweep net sampling, what you usually need is your net, of course, a container, ethyl acetate, and I should mention that maybe like cotton or some sort of cotton ball or a piece of cloth and a paintbrush. The ethyl acetate is a chemical that helps to euthanize. Who's on mute? Wants to say something? Okay, I think someone just joined. Okay. So ethyl acetate is one of the ways that you euthanize the organisms, because you don't want them to suffocate to death in the container or just be uncomfortable. What you can do is when you put the ethyl acetate, the fumes from it kind of knocks them out, and then over time they die without like suffering a painful death. So it's a way to humanely euthanize the insects. Okay, because insects, I guess I don't know if they feel pain, but they definitely can feel discomfort. So it's one of the ways that you have to be gentle with them. Paintbrush, because you have to be gentle with these organisms. You can't exactly, like sometimes you might want to hold it, and before you know it kind of crushed in your hand. So that's why you have to kind of like use a paintbrush to kind of gently pry it out of the net into the container. Kick netting. So we're moving on to kick net sampling. So usually for kick nets, you need the net, the actual net. You need waders if you're in cool water. So we work in the Caribbean, we usually just enter the water like that and then dry off after. So if you realize this is called a wader, so it kind of covers you so you don't get soaked in the water. And this is especially important because if the water is cool, because this person is probably sampling in a temperate region, if the water is cold, then it is ideal to have waders. You have to get a container, same thing, a paintbrush and some ethyl acetate or formalin to preserve the specimen. So if you realize this is a crab and there are some other little benthic macro invertebrates as well. Okay, so this is mainly, and the reason why I introduced entomology is that a lot of what we collect from a lot of these sampling methods are insects. So even in the water, in the fresh water, we get benthic macroinvertebrates, which are like freshwater insects as well. Quadratic sampling, you can use quadratic sampling for anything. Trust me, you would be surprised how much you can use quadratic sampling for terrestrial ecosystems or aquatic, because this here is a field of grass and this here is a seagrass bed. So the photo isn't as clear, but this is actually underwater. This is an underwater photo of a seagrass bed. So sometimes when we sample, I remember one of the labs at UE, when we go to Discovery Bay, we take a quadrat about this size for the one that's in the field of grass. We take this quadrat and we put it on a rocky shore. So the rocky shores, like, there are some shores that have like sand, and then there are some shores that have like mainly like rocks, especially when you go to Jamaica, has a lot of dry limestone. So in that zone, we realize that there are different snails inhabiting the shore at the different, further away from the beach. So closer to the water, you get one species of snail or one or two species of snail that you realize. Then when you push the quadrat back a little more, you realize you get another species of snail. And then in another zone, you get another species of snail, right? And because these snails are adapted, some are adapted to, live closer to the rocks that have a lot of water coming in. And some are adapted to more drier ecosystems, but close enough to the shore that if they need water, if they need to get to water, they can get it. So what we did, you all see in my cursor, well, I hope you are, because I'm showing you that I think what they did is that they decided that they are going to sample 1, 2, 3, 4, 5. So they look to see if any snails are in this section, and then they choose a random block. So so in this instance, they might say 1, 2, 3, 4, 5, and then they might choose this one. And in another time, they might choose this one. Same thing with the seagrass beds. We throw the quadrat and we see, okay, are we seeing sea snails in it? Are we seeing sea cucumbers? Because when we threw the quadrat, when we were doing the marine lab for the ecology trip, we remember seeing that we are seeing, sea cucumbers. So we drop the quadrat on a seagrass bed and we see how many, how much percentage cover. So if you can see, so roughly this section, I would say about 60%, roughly more around 75% of this quadrat has seagrass cover. Okay, so that's how you usually check it out when you're sampling. Transect sampling. So let me see if I can zoom in. This here, a transect is like, you have a measuring tape when you're trying to like build a house. So think of a transect as a long measuring tape, which it is, and you can use it under the sea. What this guy is doing, he is trying. So we have done this as well for my third year lab at uni. What you do is you drop the transect and you swim along the transect line, if it's like 10 meters or something, and you swim along that transect line and you record at each point. So every 10, so if you're doing 10 meters and every 1 meter interval, Okay, so at this, if this is a 1 meter interval, you observe this type of coral. This type of coral, what's the name of it again? It's the mustard coral, mustard hill coral. Because if you realize the color is kind of like the color of mustard, and it has like these kind of undulating hills, so it's called the mustard hill coral. So he will know, okay, well, at 5 meters, there's the mustard hill coral. At another, at 6 meters, the mustard hill coral. This here at this time is just coral rock or coral vegetation or algae. So you go along and you measure it. This here, this thick, what happens is that we also swim on either side of the transect, and we look for urchins. So when we swim on either side of the transect, we look to see how many urchins are there, because remember I was telling you all earlier when we spoke about coral reefs, how important urchins are to the health of the corals. So when we swim along and we see, okay, well, this section of the coral reef tends to have more urchins than the another section of a coral reef or another coral reef altogether, we realize that, okay, well, the presence of urchins tend to ensure and increase the number of corals. So we realize that on one reef, when we did the research, we will realize, okay, on one reef, you get more corals because there are also more urchins. But on the other reef, there aren't as many urchins, so you have more algae and other type of vegetation. I think this is it mainly. Let me see, I think this is it for the session, for this PowerPoint. So I am going to open Word now. And anyone has any questions about the sampling? Anyone has any questions? Everyone's okay. All right, I say I did the exact time here. It's 1:45. So I'm going to open Word and then I'll share it. I want us to start, you all can write it though. You all can write it on a sheet of paper. The methodology. So let me go share this document. Okay, so I want to do the methodology for the assignment you'll have. Let me open the assignment. Okay. You all see in the report? The instructions for tropical forest. So the virtual insect box. For this virtual insect box, we're choosing the location. So site description, you're just going to explain the site description. But for the methodology.
Excuse me, Miss. I don't
Yes,
know if it's supposed to be seeing other things, but I am seeing that's the word methodology.
Okay, all right. So y'all not seeing what I'm seeing. So I just needed to refer it. So if y'all could open, new share. Let me share this. You see it? No?
Yes,
Yes,
Okay. So, okay, I'll just add to this. And so you see the list of items you need. So I would start items needed. What we need. You'll call it out for me. I'm giving you all the answers are fine, man.
The ethyl acetate?
Ethyl acetate, yeah. What else we need? We need piece of cloth or cotton. Right. I think that's it for now. Yeah. All right. So this is the items. So the procedure. So I want you all to write it in past tense though. So the procedure. So what's the first thing you do? Select a field and select a location. So in this methodology, you are going to put, okay, let's say you, what field you would select. Okay, I'm going to select the botanical gardens. So I would say something like the botanical gardens. Next, so the Emperor Valley Zoo was selected because of its because of its diversity. Let me scroll up a bit. It's diversity of plants. As such, I expected it. So, well, you can say some of this, but I think this will probably go into more the site description. Okay, so I'm gonna move this into site description. So I would say I feel like this kind of falls more into the site description. Okay, so this is just an example. Okay. All right. I want you all to write it. I'm not gonna repost it. I'm leaving the lab at this document that's on the e-Biome site, just as it is, eh? I'm just showing you all. So set a location example. So you'll see the botanical gardens was the location I selected. It's next to the Emperor Valley Zoo, and it has a high diversity of plants and insects. And you move on because you already explained that in the site description. Sweet net. Okay. And then we're gonna say we sweep the net. And okay, so okay. So you see the methodology is pretty short and it's very straight to the point. So I just want you to explain that which location you chose, why you choose it. Well, that would be for the site description on why you choose it. But then you just mentioned, so you go to your site and then you use your select a section. So the Exora, I should put that in capital letters. Exora hedge, Exora, those are like the little pretty colored bushes. And you kind of just, so you're seeing that you swept the neck back and forth for 10 seconds, and whatever specimens were collected, so you sweep it rigorously for 10 seconds. So you're counting 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and you're going back and forth quickly. And you put it in a jar soaked in ethyl acetate. After that, you okay. So you all have this written down more or less, because I might be away finishing soon. I just wanted to show you this, how you write the procedure. Okay. So I expect that. And as I said, it's not long. It's literally not long. You just have to talk about the insects you select specifically. So if you get 8 insects, if you get 8 or 10, but if you can get 8 insects and explain just, I say 3 to 5 lines, but it could be 2 to 4 if I'm feeling nice, 2 to 4 lines. But I just want to see that you have an idea of how you go out in the field sampling, because, and trust me, whenever, you know, I don't know about Jamaica, but I know Trinidad has some really strict protocols right now. But nevertheless, whenever you are able to, if you can get your family to carry you to do the same set of sampling, I think you would really enjoy it. I think you would really enjoy it, because it's super, super simple. So I said 3 to 4 lines. So let me change that. I may re-upload it. I'm not sure yet. 2 to 4 lines. Okay. So here you see it again, right in the discussion, 2 to 4 sentences about each insect that you have chosen. I think I showed you all how to do the insect box. So if you could get the images, line it up. If you're lining up 5 per page and then 5 on another page, or 4 on one page, 4 on another page, you can do that. Okay. So before we go, let me stop sharing. Pause share. Stop share. And let me know if you all have anything, what you all thought about the session today. Any things that you all want to do in the time coming up. Talk to me, tell me.
Miss, I liked today's session a lot. I find it was very interactive. I liked it a lot.
That's great. That's great. I'm glad you enjoyed it. And yeah, and as I said, you all can, I hope once somebody decided, I hope a few of you at least decided that, terrariums are a fun way. So if your parents might not allow you to have an aquarium or a pet right now, I say you just start to collect your glass jars and start to make some terrariums. So you could still have a piece of nature in your room or in whatever section you have, or whatever, by your study desk or something, okay? So I think I'm more or less finished for this session. I'm going to stop recording now. If you all have any questions. So Miss, okay, hold on, let me see. Oh, okay, so Shanice is saying she didn't get to see. So let me show you an idea. So let's say I'm going to share a new blank document. Just hold on a second. Okay, so I'm going to go home. Let me share the screen now. This blank document. I was saying, Shanice, that you could do, so if I'm going to do the layout, I can say from this point forward. So I want it in letter size and the orientation to be landscape. And I'm going to put insert. No, I don't want to add, it might stop the program. So in the meantime, I'm going to look for some photos. Let me copy some photos that we just used. Copy these. Copy. Right? So then let me open it up. Just a second, I want to go back to full screen. All right. So you can make this image smaller, make these images smaller. You're seeing Shanice? Okay. And I'm gonna add a caption to this. Insert caption. Net. So I'm gonna say, so it's figure 1, okay. I'm gonna put net. Right? And sometimes you're able to section it out. You could maybe use PowerPoint if you know how to use it, but if not, don't be too hard on yourself. But I just want to show it. I want you all to see how the insect box more or less. And I want you to add, is it design? Page borders. So when you add the borders, so you could do like a 3D box. Let me see that. So, or you could select it for the particular page. So you see, you have the idea of a box, but these photos will be of insects instead. You understand? So that's just kind of more or less what we did. So I'm gonna stop sharing now. And as I say, you could get, if you could get.
Excuse me,
8.
Miss.
Yeah, sure.
I have
Okay,
a
finish up.
question.
Mhm.
Next is like how you had figure 1 and then figure 2 right underneath it. Right
Mhm.
next to figure 1. Will you put the information about whatever insect you choose, or will you put another image?
I was hoping for you all to do the image by itself, but as I said, I think because it's virtual, I think we can allow some